All right, everyone. Hello. This is the lesson about transcription and also about how we process RNA. It's going to be really helpful for you while you listen to this video to have the printed copy of the notes out or to have a piece of paper out and kind of jot some stuff down. I'll be making some notes as we go. And so I want you to be prepared for that. So if you need a moment to get out your printed notes packet, go ahead and do that now. Just pause the video. And here we go. All right, so this is just a quick overview about proteins. This really hails from like Unit 1 about what proteins are. They are polypeptides. Anytime you hear the word peptide, you need to think protein, okay? So we know, again, from Unit 1 that they're made up of amino acids. Those amino acids are held together by a peptide bond. Remember, peptide, protein. Think of the same thing. So those peptide bonds are just the bonds that connect amino acids together that make proteins. Okay. Now the real star of the show here is this term right here for gene expression. So the next two kind of lessons that we're going to talk about is how do we get proteins in the body? How are those expressed? And what does that process look like? So we call this oftentimes protein synthesis. And it kind of happens in two steps or two stages. We have transcription and translation. These are things that that you do talk about in baby bio. But of course, we're going to go into a lot more detail here about what's happening in this process. Now, I want to direct you to the picture that's down here. This right here, this is worth, I think, writing down somewhere. This whole idea of starting with DNA and then going to RNA and then RNA to proteins, we call this the central dogma of biology. And a central dogma is like a widely held set of beliefs, something that a big group has agreed upon to be true. And so the central dogma of biology is that we're always going, in order to get proteins, we start with DNA, then we go to RNA, then from RNA to proteins. The process of going from DNA to RNA is called transcription, and the process of going from RNA to proteins is called translation. This lesson is going to focus on transcription. Okay, so the process of transcription is when we go from DNA to RNA. This process is going to happen in the nucleus because DNA doesn't leave the nucleus. So in this picture right here, you can see that this is right here double-stranded. This is DNA. And then we're going to turn it into mRNA, messenger RNA. And that will be able to leave the nucleus. And so that's why we have to have that mRNA. Now, translation is when we take this mRNA, it leaves the nucleus, and then it's going to find a ribosome, and the process of translation will happen where it builds the protein based on the mRNA sequence. This happens at the ribosome. So transcription happens in the nucleus, and translation happens at a ribosome. All right, so it is going to be important that we understand the three types of RNA as we move forward. I'm going to go ahead and kind of zoom through these. So we have messenger RNA, or what we also call mRNA. And this is what is made during transcription. We use a DNA template, and then the process kind of reads the DNA sequence that's there and creates complementary RNA bases. So don't forget that the RNA bases A pairs with U, not T. And then the mRNA is going to carry this information from the DNA at the nucleus to the ribosome in the cytoplasm. So messenger RNA, it's a messenger. It's taking it from the nucleus and out to the ribosome. Now, transfer RNA molecules, this is going to be more important in translation. But tRNA, or transfer RNA, kind of looks like the shape of a T, right? So the most important part that you need to know here is down here at the bottom. Down here at the bottom, each tRNA molecule will have something that we call an anticodon. Now, a codon is a set of 3 letters, right, that mRNA has. So every 3 letters is a codon. tRNA has a complementary one, and it's called an anticodon. Okay, so those are going to kind of match up with each other. And then this is going to be what kind of actually makes the amino acid that gets added to the protein chain. And then we have rRNA. This one is very brief. It essentially helps make ribosomes, and it helps link amino acids together. You're not going to see this one quite as much as the other two. All right, so here's really what we call the codon, right? It's a triplet code, which means it's a set of 3 letters. During transcription, this bullet point right here is important. Only one DNA strand is being transcribed at a time. This DNA strand is known as the template strand. It also has other names: non-coding strand, minus strand, antisense strand. But really you can think of it as the template strand, because that's what it's going to read in order to create the mRNA. Now, the mRNA molecules are also anti-parallel, and they are complementary to the DNA nucleotides. So, for example, the complementary mRNA base is going to be U, T, A, U. So think about your complementary base pairings, right? And then every set of 3 is called a codon. That's what we were talking about. So this right here, whoop, 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 that's one codon. This is also a codon. This is also a codon. So these are the sets of 3. These codons are kind of what determines which protein gets built, or which amino acid gets built. So the codons code for amino acids. This is this part right here. Now, just for your information, there are 64 different codon combinations. 61 of them actually do code for amino acids. 3 of them are stop codons. Stop codons are a message to all the parts of the cell that something needs to stop, right? And that's why they're called stop codons. Amino acids and codon combinations are also universal to all life. So that means anything that's alive, that's living, is going to have these 64 different codon combinations. Now, this word redundancy, in life, redundancy means that you repeat yourself. In biology, what it means is that we can have like more than one codon. So remember, a set of 3, more than one codon that can code for the same amino acid. So for example, this one right here, VAL, I think it's valine, but VAL for short is our amino acid. If you notice, all of these combinations right here code for the same amino acid. So even though they have different letters, they're coding for the same amino acid. That's what we call redundancy. Now, your reading frame, think of it kind of like you have to count your codons in the right order so that you can know exactly which amino acid you're coding for. So they have to be read in the right grouping. So, for example, the fat cat ate the rat is a sentence, right? But if you switch your reading frame a little bit, it's not going to make a ton of sense. So for example, here's your little example bullet point down here. If the reading frame shifts even by 1 letter, it will produce a completely different outcome, right? And so this is our result here if we kind of shift these things. Okay, these are the answers to the quick check. If you'd like to write them down in your notes, please do so. Now we're going to really talk about the steps of transcription. And so this is the part where you need to make sure that you're listening, paying attention, pause the video or go backwards to listen to it again, because this is going to be the main place where you're going to get the information about what's actually happening. Okay, so there are 3 general steps in transcription. We have initiation, elongation, and termination. So before we get into like the actual steps, let's talk about what these 3 words mean. Initiation, of course, we know that means to start, right? Elongation means something gets bigger. And termination means something stops. And that's kind of very loosely what's going to be happening in these 3 steps. So the first step is initiation. Remember, initiate means to start. And so transcription is going to begin when RNA polymerase, there's that guy again, RNA polymerase molecules are going to attach to something called a promoter region in your DNA. Here's what a promoter region is. So you have so much DNA, right? Like the human genome is just enormous. And a lot of the DNA that's present actually doesn't really code for anything, right? And so how does your body know, how do your cells know where to transcribe a gene? So for example, here's your DNA, right? And then let's say that the gene is only right here. Okay, so you've got all this other DNA that's happening that doesn't mean anything, but this area right here is the gene. How does your body know, how do your cells know where to find this? Well, that's where the promoter region comes in. So promoter region is going to happen kind of before the gene. And so what it does is it's a signal that says, hey, RNA polymerase, come bind right here, because everything after here is the gene that you need to transcribe. So that's kind of what a promoter region is. Here, for eukaryotes and prokaryotes, it works a little different. They both have promoter regions, but in eukaryotes, which is you, right, you have to have this thing called a Tata box. And a Tata box is a sequence of thymine and adenine that kind of sits here in the promoter region right here. And that is where the transcription factors or RNA polymerase is going to bind. Prokaryotes, things like bacteria, they don't actually need the Tata box. The RNA polymerase can actually just bind directly to the promoter. So here in eukaryotes, like in your genes, you have to have this little box, and this is kind of a signal to tell the cell, hey, this is the promoter region, this is where things are going to start. And down here at the bottom it does say, look that the promoter region for both is going to be upstream or come before where transcription actually starts. Now, the next step is elongation. And this is the process of kind of building. So RNA polymerase is going to open up that DNA strand. It's going to read the triplet code. So remember, those are codons of the template strand. It's going to move in the 3' to 5' direction. And it says the mRNA transcript elongates 5' to 3'. So if you notice here down at the bottom, it says elongates 5' to 3'. So this is kind of like in DNA replication, right? DNA polymerase moves 3 to 5, but your new strand is built 5 to 3. Same thing here. RNA polymerase is going to move 3 to 5, but the new mRNA strand right here is being built 5' to 3'. RNA polymerase is going to move downstream. In this process, they're only going to open small sections of DNA at a time. Of course, don't forget about our complementary base pairing rules. So for RNA, A is going to be with U, and C and G are always together. Okay, and then the RNA strand is going to kind of grow away from the DNA so it can be its own thing. Then after it's done, the DNA double helix is going to reform itself, which is great. A single gene can be transcribed simultaneously by several RNA polymerase molecules. So this means that it can work faster, and we can make proteins faster in our body, which is something that happens all the time. Our last step in transcription is called termination. So in prokaryotes, there's something called a termination sequence. So remember, prokaryotes, this is things like bacteria. And this termination sequence that happens, what it's going to do is it's going to basically give a stop signal, a termination signal. This is when the RNA polymerase is then going to detach itself. The mRNA is released, and then it can move out of the nucleus to go to the ribosome to go on to translation. Now, just so you know, mRNA does not need modifications. It's just reading the DNA template and creating those complementary base sequences. In eukaryotes, though, RNA polymerase is going to create a sequence of DNA called the polyadenylation signal sequence. So the polyadenylation signal is this guy right here. I would memorize that. AAU, AAA. This is going to release the pre-mRNA from the DNA, and then it has to do some modifications. So in prokaryotes, we have a termination sequence. And no modifications. And for eukaryotes like you, we have a polyadenylation signal sequence, which is AAUAAA, and modifications must be made. So if you're like, well, what kind of modifications have to be made? Well, there are really three that have to happen before the pre-mRNA is ready for translation. So this is a 5' cap, a poly A tail, and then RNA splicing. We're going to go through each of these and make sure you take notes as we go. So we have a 5' cap. So essentially at the end of the pre-mRNA, it needs a cap. So it creates this like sequence of modified guanine, so G, and it's going to put that end on the 5' end of the mRNA. Then we need a poly A tail. So this happens on the 3' end of the pre-mRNA, and it's going to receive somewhere between like 50 to 250 adenine nucleotides. So poly A tail, adenine. Poly means many. A, adenine, so it has many adenines that are kind of added on to the end of the 3' end. So the 5' end gets a cap. The poly A, or the 3' end gets a poly A tail. So why do we need those though? So this is going to help the actual mRNA leave the nucleus. It's going to protect the mRNA from degradation. This means to break down. So DNA is somewhat fragile. So to help with the movement and to keep the actual important coding information from getting broken off, they add the cap and the tail to kind of give a little bit of a buffer room. Then the other purpose is it's to help the ribosomes attach to the 5' end of the mRNA when it gets to the cytoplasm/ribosome, because that's where the ribosomes are. Okay, so here's your 5' cap right here. So this is on the 5' end of mRNA. Then you have your poly A tail. This is on your 3' end. And all of this is to help essentially protect the mRNA and help get it out of the nucleus. Now, RNA splicing is something that is very interesting. We have sections of the pre-mRNA. We call them introns. Those get removed, and then exons are joined together. So think of it like taking out the non-important information. So introns, it says intervening sequence, do not code for amino acids. So these introns, we don't actually really need them. And if you're like, well, why are they there? They're there to help give the mRNA some options when it comes to what protein it codes for. So we have introns, and then we have exons. Exons, these are the expressed sections. What that means is they code for amino acids. So if you look at this picture down here, this first one right here, we have exon, intron, back and forth, okay? Then what's going to happen is we're going to process that RNA, the pre-mRNA. We're going to take out these introns right here, and then we're only left with our exons, which are the actual parts that code for an amino acid. So introns, think of it like a placeholder. And your exons are the actual information. Okay? So splicing something means to kind of cut it up or cut it apart. And so that's what's going to happen, is it's going to kind of cut things out, and we'll see on the next slide. So why do we splice the DNA? So a single gene can code for more than one kind of polypeptide or protein. We call this alternative splicing. So we can make different combinations of proteins based on the introns and exons that are left, or the exons that are left. So, for example, we can splice this gene this way. This is the first way right here. We get protein A. It's in this order, 1, 2, 3, 4, 5. The second way we can do it is we can just do 1, 2, 4, 5 and leave out 3. Then over here, our third way, same gene, 1, 2, 3, 5, so we're missing 4. This is just an example of kind of what your genes do. We can take the gene, and because if it codes for more than one type of protein, then your body needs to be able to decide which protein it's going to use. This is what alternative splicing is. So think of it as cutting up the pieces and recombining them to make different products. Okay, so I'm going to do a quick overview of all the things we've kind of talked about, starting with just the process of transcription and kind of what that looks like, and then also about the alternative splicing. So this first picture right here, let me make this one little. This first picture right here, we have our DNA strand, right? We have something, this picture is from Khan Academy. It's labeled as, it has a coding strand right here. Don't worry about that right now. I'll get to that in a second. But essentially, RNA polymerase comes through. Now, let's assume that this is a eukaryotic cell, right, with the DNA. That means if RNA polymerase is here, okay, then it had to have a like a promoter region, and that's going to be right here. Okay? RNA polymerase is going to bind. It's going to open up the DNA strand, and then it's going to just synthesize one strand right here. This strand that it's using as a template is called the template strand, right? So it'll go in, RNA polymerase goes in, it reads the DNA strand and creates a copy of it, but using RNA bases, complementary RNA bases. So in this box down here, okay, here's what we have. We have the coding strand and the template strand. These are our original DNA. But RNA polymerase is only going to synthesize one strand at a time using the template strand. So you can see here, here's our RNA. It's got our RNA bases, A, U, G, A, U, C, because it's complementary. Complementary. And don't forget, we're using U because these are RNA bases, not DNA. Now, something to notice about the coding strand. The coding strand is the other side of the DNA that's not really being used. And if you notice, these are essentially the RNA, the mRNA that's created, and the coding strand are essentially the same. The only difference is it has RNA bases instead of DNA bases. So RNA polymerase comes in, opens up the DNA strand, reads the template strand, and creates that mRNA from that. Now, once we're through with that, we go into splicing, right? So here is our gene, our DNA. We have our promoter region right here, okay? And then exons and introns are present. Remember, introns, those are the ones that don't really matter. They don't really code for anything. So we can do some alternative splicing, and we can take out these introns that don't really matter. And now we have mRNA that has been spliced that then can be used at a ribosome to make a protein. Hopefully this makes sense. Now, once all the modifications have happened, the pre-mRNA is now considered mature mRNA. It can leave the nucleus, it can go to the cytoplasm, and when we say cytoplasm, really what we're referring to is the ribosomes that are in the cytoplasm, and that's where translation happens. I really hope that this video helped you guys. This is the quick check, so if you want to write down these answers, you can do that at any point. But if you have any more questions, make sure you write down some questions to bring to me about the content that I covered here.